Calcutta. So today I will be talking about Chitala, a new candidate species for aquaculture diversification. This is not a new species, but from the farming practice, we can consider these as the new species for aquaculture diversification. So that's why I have chosen this. So that was also my part of the PhD program, and so I will be talking about this regarding this species, that how we can introduce this species as a new candidate one for aquaculture diversification. So let's start with. Can you see my screen?
Yes.
Yeah, fine. So 20 years ago, Nature actually published a review characterizing aquaculture as a possible solution and contributing the factor to declined fishery stock worldwide. Despite impressive gains, aquaculture sector still faces serious challenges, in some cases undermine its ability to achieve sustainable outcomes. Hence, it is considered as the last resort for endangered or the threatened fishes. In this slide, I have just tried to ask the questions that what challenges do you face with the unprecedented human growth in aquaculture? Why we have not still reached the sustainable developmental goals in aquaculture? And how can we reach the sustainable developmental goals in terms of aquaculture nutrition perspective? We will try to answer this with my lecture, but based on these questions, I have framed the roadmap to give you an understanding on the sustainable developmental goals and its role of aquaculture, conventional dilemma of nutrition. That means when you are trying to feed a fish in a large scale basis, what conventional problems do you face? And what are the alternative trade-offs? That means when you are using some alternative ingredients, so what will be the cost benefit ratio or the trade-offs you generally face? And the stages of nutrient acquisition, that means how the fish they acquire their nutrition during their development or through their ontogeny. Temporal pattern of acquisition, that means it is a timely basis, that means during their developmental time how they acquire their nutrition. And lastly, understanding the temporal pattern of nutrition acquisition in new candidate species, that is Chitala. So as a global society, we are around 7.5 billion people and we are heading towards 9.5 billion by 2050. Not only does this present the problem of an additional 2 billion mouths to feed, human diets are also increasingly shifting in line with the growing prosperity. And the sustainable aquaculture is a dynamic concept, as you know maybe, and it depends on the species, location, societal norms, state of knowledge and technology. All this should be aligned with altruistic necessity for achieving the goals. So sustainable use of a new species or the candidate species with its stock enhancement and systematic conservation, it needs a good scientific understanding on their biological attributes and cultural potentialities. Moving on to the next slide, that is what is sustainable aquaculture. As I have already mentioned that sustainable aquaculture is something that all of the stakeholders, so starting from the farmer to the owners or farm owners or who are selling the fishes, that means in the entire value chain, they claim to support, but their institutional mandates and incentives are not always aligned with the altruism necessary for balancing the goals of these pillar of sustainability. So what do we mean actually based on this sustainable aquaculture? the sustainable aquaculture are based on these 5 pillars. First one is the environmental, second one is the technological, third one is the economic, fourth the demographic, and last but not the least the governance and the policy. So there are some decisive factors that for the successful freshwater aquaculture, as I have selected this species which is a freshwater one. So I have just pointed out the decisive factors for successful freshwater aquaculture. These are the environmental degradation, feed, freshwater resources, collection of the wild juveniles, and the mortality rates. But among these, the feed is one of the most important feature or the factors which actually decide that how the fish will actually grow. They will grow faster, and what will be their requirement during their temporal acquisition. In our lecture, I have concised my discussion mainly based on this feed. As we all know, there are 17 sustainable developmental goals which was proposed by United Nations, right? So among these goals, the aquaculture being the fastest animal producing sector, it can promise to achieve these sustainable developmental goals by offering sustainable food system. Here we will try to figure out two main aspects of aquaculture, that is the fish feed and the culturable species and their role to achieve the sustainable developmental goals. As we all know that the diversity of the species created local social benefits to the small scale farmers, offering both biological and economic benefits in aquaculture. So dependence on the wild fish stock widely questioned in the recent years. But there are some stumbling blocks when you try to design the food or we try to design to make a proper diet for a particular species. As because the fluctuating state of fishery resources are in the fishing zones, there are overexploitation or exploitation of the fish stocks which we actually used as a conventional meal and we use them as the fish meal. Climate change and phenomena like the El Nino and the introduction of the fishing quotas along with the increasing pressure to use fish oils and fish meal in other sectors like the cosmetic industry or the medicine like that. So these are in contrast, these are some recommendations for achieving the sustainability in terms of alternate aquafeed with probable recommendation for achieving sustainability. So I have here shown that these main 5 or 6 pillars which actually define the sustainability of aquaculture, I have made the probable recommendation to achieve the sustainability and it is already been published. Now the question is where we stand. That means to manage the feeding practices, we need to understand the importance of feeding in the fishes. So there are some definite issues to achieve the sustainability. That means availability and cost of the feed resource to develop the alternate aquafeeds, increasing competition of raw materials in the resource with the other sectors, demand supply forestalling of the local and global market in consort with the maintenance of environmental quality, social acceptability and economic growth of aquaculture systems, and the reduce the overexploitation and choosing the right species and to manage the feeding practices. So up to this we have focused or we have discussed already that what are the sustainable developmental goals and why the feeding are the most important part to reach these sustainable developmental goals in terms of culturing a species and the local species which actually can give upliftment in culture to get an additional input in the aquaculture society. As we all know, these are the importance of feeding in fish. That means it must eat to survive. Size often influences susceptibility to the predators. Fish never stop growing, and maturity is a function of size. More food, more energy for growth and reproduction, higher foraging rate, higher fitness. So what are the food types or the components and the diets? This is one of the important challenge for being an aquaculturist. You have to consider the natural feeding habit of the fishes. That means the animal's ecological interaction to its behavioral and physiological adaptation should be in synchrony. Otherwise, you will not be able to design a particular aqua feed. There are different types of the fishes, as we know, that they are termed as the herbivore, carnivore, detritivore and omnivore. So at a finer scale, fishes can be divided on the basis of their feeding yield. That means either they are the planktivore or they are the herbivores, like that. Okay. So when you got the information that particular species is herbivore or carnivore or a higher resolution, in which guild they are belong to, you can try to understand that what are the stages of their nutrient acquisition. That means how the fish detect their food, and when they got that food, how they process it. Either that will be processed mechanically or chemically or microbially. After that, there will be the physiological and integration of the sensory modalities by which they actually utilize or absorb these material what they have got. So the nutrient intake that can be in the next level can be utilized as a digestive product or the absorptive one, and that will be distributed and ultimately metabolized and excreted by the fishes, and the nutrient macromolecular interactions will be there, and the fish will try to grow and fish will become bigger and they try to reproduce. So this is the utilization of a nutrient in the production of a physiological response. Now, what do you mean by the temporal pattern of nutrient acquisition? Temporal, the term has been derived from the time. So, during the time of development, in better way, we said it as a ontogenic development. So, ontogenetically, how they are getting their feed, that means we can divide this temporal pattern in three different ways. First one is the daily pattern, that means how the fishes, they are utilizing their feed in a daily way, or the seasonal pattern and through their developmental period. So daily pattern, that means it involves two integrated phases, times of feeding episodes and the rates that define this daily pattern, and rate at which the ingesta they travel through the alimentary tract and through digestion and absorption. And during the seasonal pattern, if you try to study that, the influence through the temperature or the food availability or the reproductive cycle, you have to consider these factors. While during the developmental patterns or the ontogenetic patterns, the diet shifts between the juveniles and the adults, and they are often associated with the changes in relative gut length and the digestive enzyme. So when they are trying to grow, I will show you that how they actually shift their feeding habit. Okay. So during this discussion, you will later find it that how they develop different kind of digestive enzyme during their development. Now we come to the species of my interest, that is Chitala, or commonly we call this as the featherback. Okay. So Chitala represents an appreciated fishery resources in Southeast Asia and particularly in India. Apart from the following aspect, it is high in nutritive value. When we analyze the nutrient and the fatty acid level in Chitala, we found that this can be a very good competitor. So I will show you, I don't know this it will work or not. There is a hyperlink. No, it's not working. So it can be a good competitor of our regular fish species. But due to the overexploitation, this species has been categorized as endangered. Thus, the large-scale farming of these species has been used or ensured the biodiversity conservation, widening the consumer choice by freshwater aquaculture diversification. So during my PhD dissertation, I did all this work, and before doing this work, actually when we tried to study that what the previously few researchers has done, we found that this work was done previously, and there are still some gaps that will remain, and we identified those gaps to study further. So the relative and detailed sequence of morphological and morphometric characters during early life stages under the culture condition that was not previously done, and the basic information on the organogenesis of larval digestive system, appearance of key digestive enzymes, composition of bacterial communities associated with the gastrointestinal tract, and we found that there is urgent to develop a formulated feed or the diet for sustaining the growth by substituting the live prey. Based on this gap, we generated few questions. That was actually our objective was that what are the developmental priorities during the ontogeny? Is it possible to coordinate the waning time based on these growth priorities? Is there any comparative indicator which will define the developmental landmarks during the digestive activities? Is it possible to formulate the diet based on the knowledge of autochthonous gut microflora to increase the feed utilization and growth efficiency? And from based on this question, we divided our objectives on the following points. So let's see. So these were our objective. We actually planned based on their that questions. So these were our objectives. The first one was the comprehensive staging series during early development with the changes in morphological features and morphometric ratios. These will help us to understand the phenotypic plasticity and the size related adaptation during their development, to understand the growth priorities during ontogenetic time scale. Our second objective was to understand the gut histomorphological changes and developmental pattern of key digestive enzyme during the early life history stages. This actually enable us to synchronize the maturation of digestive organ with the feeding protocol and formulation of cost-effective age-specific diet. And our last objective was to explore the intestinal or the autochthonous bacterial flora and their screening for the evaluation as a probiotic supplement in the diet. They will help us to identify the resident intestinal bacterial flora, to understand the potential of the gut isolated strain for the feed utilization, growth and survivorship of the juveniles. And ultimately we wanted to get a bigger picture, that means holistically they will help us to understand the optimization and refinement of the present rearing protocol for effective culture practices. So what we actually tried in a nutshell, that we wanted to design a feed. So until and unless we understand the basic physiology or the basic growth pattern of that particular species, we will not be able to understand or define or to make a feed that is age appropriate. So ultimately if we can make the feed, we can try to get a bigger picture to make the intensive culture of this species. And as we know For the intensive culture, we have to give the supplementary feed, or we need to make some feed with the additives, right? So now I will tell you about my first objective and how actually I accomplished that. The comprehensive staging series during the early development with the changes in morphological features and morphometric ratios of the featherback. This was the materials and methods for the first objective we have followed. Artificial breeding was performed and the eggs were incubated in the circulatory system. As you can see, this is the circulatory system, and the morphometric characters of the following stages of fishes were done, and it was analyzed with the allometric growth model and inflection point based on these references. Now the result. So what we actually found that 4 developmental stages were observed. One is the yolk sac stage, second is the preflection, third one is the flexion, and fourth one is the post-flexion stage. So these were the stages we found during their early development. So mouth and anus opened on the 8th day after hatch, as you can see from this picture, and lens and the buccal aperture that opened, and swim bladder inflation and aperture or the apparatus for the teeth on the 10th day occurred. Yolk sac reabsorbed and exogenous feeding along with the fin fold difference started on the 12th day. So 12th day was a important landmark for this species. And on 15th day, appearance of the fin rays that was observed. So first appearance of the juvenile pigmentation or the chromatophores you can see in this species on the 25th day after hatch. That means when that fish they come out of hatching. So from that time to the 25th day after hatch, you will find this. So this is the total length with respect to the day after hatch showed the linear relationship. The total length reached 30.45 millimeter at the 30 day. Okay, 30 day after hatch. During the yolk sac phase, there was very low increase in the total length. These are the graphs representing the allometric growth equation and relation between the different measured body proportion along with the total length. So we have taken the ratio between the total length and the other different body parts. As you can see, the tail length or the body length or the head length, like that. Here the equation followed was y is equal to ax to the power b, where if b is greater than 1, it will be considered as positive allometry, and if it is less than 1, it was negative allometry, and if it is equals to 1, then it will be isometry. So these are the results, and we have make it in a simpler table where we found that head length, tail length and the tail depth, they showed the triphasic growth. That means it showed 3 different developmental phases, and the head and the eye diameter depth, they showed a biphasic growth. So snout length and the body depth of pectoral fin and the mouth gap, they showed biphasic growth, and the TRL that showed the triphasic growth. So during developmental period, the head length, tail length and the tail depth, they showed positive allometry at the first 2 phases followed by the isometric. But in case of the head depth, pectoral fin and the eye depth or the snout length, they showed positive allometry followed by the isometry. And for the mouth gap, you will find that this is the mouth gap. You will find a negative allometry, then the isometry, and the TRL showed a negative allometry, then the positive one. So this is actually what we can conclude from this, that the first phase of development, they showed energy and building material allocation in priority. That means the head or the tail depth, they actually got the primary allocation in priority, where the cerebral, sensorial and suspensorial or the motor activity was important than the size increase, right? So subsequent developmental period, they characterized by the flexion of notochord, rapid acquisition of adult axial muscle distribution, and accompanied by the rapid development of the fin rays, so which help them in better swimming and prey capture. So our key findings were that the relation between total length and the age followed a linear function. The yolk sac and the preflexion stage manifested by precedence of sensorial, feeding, respiratory and swimming systems by the accelerated growth of head and tail. And the flexion stage was characterized by change in growth priorities, performing crucial function in swimming, maneuvering off for the feeding, and at the end of post-flexion, it was evident that the tendency to isometry for all the growth coefficients corresponding to transition towards the metamorphosing stage. So developmental pelvic fin was rudimentary during the entire studied period. Now moving on to our second objective, that is the gut histomorphological changes and developmental pattern of the key digestive enzymes during the early life stages. Again, this was our materials method. Here we can see that the total length and the body weight showed a linear relationship, right? So it showed a linear relationship and the mouth gave reached 2 millimeter. It reached around the 2 millimeter here, 2 millimeter, which implies that the larvae can be went onto at the end of the study. That means in the early phases, you can use the micro diet starting from 50 micron. The histological the histological section, they show that one day after hatch, the mouth was closed and an undifferentiated yolk sac surrounded by yolk sac syncytium. This histological section showed that 8 days after hatch, oral valves were open. So these are the oral valves that was opened and showing the semen gland around it. There was the glossohyal cartilage, pseudobranch and the teeth. On 10 days after hatch, the teeth were there. So you can find the teeth. Okay. So there was a transition between the esophageal and the gastric stomach. And I will show you in the next slide that there was a transition in the foraging and the feed capturing ability, which implies that the esophagus and the gastric stomach, it was surrounded initially by the yolk material, and at the 12th day after hatch, esophagus showed complete 4 layers and there was increase in the mucus cell. So you can see the increasing in mucus cell in the section. This is the histological section at the 15 day after hatch, where the stomach, it showed the lumen. You can see the lumen and the gastric pits and 3 layers. The numerous gastric glands observed on the 25 day after hatch, which indicates the larvae can digest variety of the prey item and can be shifted from live feed to the artificial feed. These are the histological section of the intestine at the different day after hatch, where we can see that at 6 day after hatch, the intestine showed the microvilli. There was an increase in the mucus cells from the 20 day. Okay, so this is the picture of the 20 day after hatch than the 12 day after hatch. So at 12 day after hatch, the pyloric ceca was developed completely, and this is the detail of the post or the post intestine and the rectum at the 12th day after hatch. This is the sagittal section of the larvae at the 8th day after hatch, showing the liver, diffuse pancreas. Okay, so pancreas is developing, And at the end, you will find there was a number of adipocyte that has been reduced. Okay, so it has been reduced, and this is the pancreatic tissue section showing exocrine pancreas and the islets of Langerhans. And at 25 day after hatch, again there will be the invasion of the adipocyte because of numerous surrounded pancreas tissue, and this is the diagram showing the gut histological development on their different days. This is the diagram where we are showing that the main histological ontogenetic landmarks during the larval development in Chitala. So 0 to 8 day after hatch, the endogenous stage was there. So this is the lecithotropic, that means they are feeding on the yolk. This lecithoexotropic, that means they are feeding yolk as well as from the food outside, and exotropic, that means the food they are taking only from the outside. So from 0 to 8 day after hatch, there was endogenous or the lecithotropic stage was observed. From 8 to 12, you can see on this scale that from 8 to 12, it was mixed with, there was a mixed phase, and from the 12th day after hatch onward, it was the exogenous phase that was observed. This is the enzymatic activity graph that is showing the enzymatic activity during the early life stages, where we can see that the specific and individual amylase activity was observed from 1 day after hatch, but from 9 day after hatch the activity becomes significantly reduced, and the initial increased level could be explained as the programmed gene expression. When we talk about the lipase activity, right? So this is the lipase activity. This is the specific activity and individual activity of the amylase. And for the lipase, this is the specific activity and the individual activity. So lipase activity was also observed from 1 day after hatch. So you can see from this graph, it can be observed from 1 day after hatch, but there was a significant increase in the activity from 9 to 12 days. These can be related to the metabolizing yolk before metamorphosis, and level in latter phase may be due to the higher efficiency to digest and utilize the dietary lipid. For the digestive enzyme, these are the protein digesting enzyme, that is the acid protease, where we can see that it was observed for the first time on the 12th day after hatch. So from starting to the 12th day, you will find no activity, and from 12th day after hatch, there was an increase, and there was an increase later with the ages. So alkaline protease first appeared on the 1 day after hatch, but after the 15th day, its activity became decreased, and this can be correlated with the development of the stomach. So when you are not having so much importance of stomach for your digestion, so acid protease will not work so efficiently. So alkaline protease may come from the hatching gland of the mother fish, and the activity of the alkaline protease before the stomach development help the larvae to compensate the acid digestion with the help of alkaline digestion. So initially the alkaline digestion was there as because the stomach was not forming at all. So initially in the gut development, there was the development of the incipient intestine, and at the later part we found that the stomach was developing. So there was a transition, right, from the alkaline protease activity to the acid protease activity, which actually defines the shift in the required species. This is the relative activity percentage of acidic and alkaline protease, showed that on 5 day after hatch, the alkaline protease activity was 100%, right? You can see from this, and but on 12 day after hatch, acid protease activity was 35%. Thereafter, in 30 day after hatch, acid protease activity becomes 60%. So this diagram shows the lipase and protease activity and their ratio during the development. So as a general rule, maximum lipase activity is related to the minimum protease activity. So during endogenous feeding period, Lipase is to protease ratio was greater than 1, but when we are going beyond, that means from the 9 day after had, it become a little higher and thereafter become less than 1. So from this histological study, what was our key findings? We need to summarize that we found that at the mouth gap width reached 2 millimeter and the proportional relationship of the early weight with the total length, this suggests that Chitala can be a good candidate for early weaning with the compound diet, because most of the cases you cannot feed the fishes at very early. Initially they take the artemia or the zooplankton, but from this we can find that when you get this fish, they have achieved the mouth gap with 2 millimeter, you can feed them exogenously. So intense histomorphogenesis observed during the first 2 stages. Stage 3 showed only increase in size, complexity and the pre-existing organs. After endogenous feeding period, amylase activity reduced but lipolytic activity increased. Okay. So less efficient intracellular and alkaline digestion was substituted by more efficient acidic digestion. As we know that the acidic protease, they break the bond in between. So we can call it as a more efficient digestive enzyme. So polynomial trend of the digestive enzyme activities suggests the adaptation towards the diet during the ontogenetic shift with the progressive appearance of digestive organs and by the response to change the available resource. So if you see the graph, you will find that these are the polynomial equation. So when you make this graph in the Excel or any kind of software system, you can easily define this equation. So from this equation, we can see this is a polynomial equation. That means there was different kind of adaptation during their entire life stages. Okay. So from that perspective, we move to our next level. That means the materials method we done to understand the major bacterial group or the autochthonous bacteria that was resides in the intestine of Chitala. Okay. So what we found? We found that, sorry, we found that there was major 9 groups of the isolates that we isolated from the intestine of Chitala, and they were namely the Bacillus, and Bacillus was the major bacterial group we found in that. Okay. So there are the major bacterial group and their isolate numbers, and from this isolate Bacillus having previously reported the probiotic potentiality, we have further characterized it. So we characterized it based on their morphological test, biochemical activities and the enzymatic activities. We done the physiological characterization, and this is the prerequisite for being the probiont, and this is the result. After the qualitative extracellular enzymatic activity performance, we found the FB11 can be a very good very good in amylase activity and very good in protease and lipase activity also. So based on this result, the molecular characterization of FB11 was done, and it showed the maximum similarity with the Bacillus licheniformis, and the strain was found to be Bacillus licheniformis FB11, we named it. So Bacillus licheniformis, and as we find it from the feeder bag, so we found it as a FB11. As the viability of the probiont is the main concern for feed development, so after analyzing the viability, we found that viability of this probiotic strain, the feed reduced on the average by 1 log unit after 90th day of experimental trial. So when you are trying to make a feed and it is not having the additive you are using, which has a very short shelf life, it is very difficult for you to use it. Okay. So the increased viability may be due to the ability to form endospore, and the reduced viability may be due to the different storage factors. So we check the water quality parameters found to be in the optimal range for the fish rearing. There is no adverse effect of the probionts on the water quality. So this is the feed utilization and digestibility study revealed that the fish feed with the diet D3. Okay, so this is the D3 diet. You see that reveal it has a highest weight gain percentage. So the highest crude protein was observed in the fish feed with the D3. So crude protein percentage was very higher, and the highest ether extract that was with the D5. The lowest crude protein was in the control, but however the probiotic concentration and duration of feeding they significantly increased the gut microflora of the species. Okay, so when we feed with the probionts, we supposed to we can expect that there was a good effort in their digestive system. So they increase or influence the gut microflora, positive gut microflora, and total heterobiotic or total heterotrophic bacteria and bacillus increase after the 14 days of the trial. And further, it increased on the 60th day. So the PPC or the presumptive pseudomonas count and the MAC or the TCC, they observed in case of the D3 at the 90th day. So the gram-negative bacteria, they were supposed to be declined, and it was also shown in this. So this may be due to the ability of bacillus to produce different antibiotics. Fishes fed with the probiotic supplement showed significant increase in digestive enzyme than the control. So after 60th day, overall activity decreased, and the highest enzymatic activity was observed in case of the fish fed with the D3. So what we found as a summary of this prebiotic or the probiotic feeding or the additive, Bacillus licheniformis AB11 can be a good probiont which can tolerate the wide range of temperature, pH, bile salt concentration, and showed very high protease and lipase activity. Viability decreased on an average 1 log unit at the completion of experimental trial period. Best growth performance feed utilization observed with the experimental diet D3. With the increased inclusion rate of bacteria, that means more than 5 into 10 to the power 6 CFU per gram, the digestibility decreased. The probiotic concentration in the diet and duration of feeding trial significantly influenced the gut microbiota, and all the water quality parameters during the entire experimental period found to be in the optimum range of fish rearing. Thus, the result of the study, we got that the growth priorities during the ontogenetic shift was essentially starting from the 12th day after hatch, and that was the landmark. And digestive enzyme was a comparative indicator of the ontogeny, and there was an establishment and appropriate weaning time, an improvement of the growth performance and the digestibility by inclusion of the Bacillus licheniformis. So thus, the result of the study will help the aquaculturists to identify limiting factors and the bottlenecks during the rearing of this species, and this will altogether help to design more suitable rearing condition for particular life stages of this new candidate species. And we can achieve the sustainable developmental goals by profit maximization with the proper feeding method, reduce negative environmental effect, increase livelihood opportunities and social acceptability with various partnership, research collaboration and awareness. So further, we can say that the future policies and programs to promote the aquaculture will require a food systems approach that examines the nutrition, equity, justice, and environmental outcomes and trade-offs across the land and the water. It is suggested to bring together into a common system available information, past and present, currently disparate and dispersed, and direct the future research towards the common goals with harmonizing the methods. Lastly, I just want to say that the more you know about the species, the more you understand about how better to protect them.